Good afternoon. We can go ahead and continue with our cancer and body imaging section. My name is Sarah Lewis. I'm an assistant professor of radiology and a junior faculty member in the Tally Lab here at tMII. And it's my distinct honor to introduce our speaker this afternoon. Dr. Olivier Gevaert is a PhD from Stanford University, and by way of background, he did his educational training in Belgium, where he has both master's degrees in electrical engineering and artificial intelligence. He received his PhD in bioinformatics. He's been at Stanford University since 2010. He is currently an assistant professor of medicine. He has a lab where his focus of his work is on biomedical data fusion. He's received numerous honors and awards for his work, including being named an honorary Fulbright Scholar of the Commission for Educational Exchange between the United States, Belgium, and Luxembourg. He has an extensive publication history with innumerable peer-reviewed publications and has a track record of successful NIH funding as well. So today we are honored to hear him speak. The topic of his talk will be multi-scale AI for modeling of complex diseases. So please join me in welcoming him.
Thank you. First, I want to thank the organizers for giving me the opportunity to present our work. So I'm going to talk about our work in multiscale modeling of cancer and how we use machine learning and AI approaches to link very heterogeneous data. So nowadays we are able to collect lots of different types of data on multiple scales, and computational analysis of complex diseases is now also possible at multiple scales because we can collect these data of molecules, cells, tissues, and patients. And this is possible thanks to technologies such as sequencing for molecular data, immunohistochemistry, and advances of pathology, and also medical images that are routinely available. But what is important about this multiscale data is that each of these different technologies essentially assesses heterogeneity in a different way. From molecular data sets, such as sequencing, we are essentially getting very deep and detailed information of a piece of tissue, but not of the lesion as a whole. For immunohistochemistry, we're getting cellular information and information about the cellular morphology of a slice of tissue. And with medical imaging, we're getting information of the tumor as a whole, and its morphology, its texture, intensity, and so on. So there's essentially a huge opportunity to study all these different types of information and different ways of assessing the heterogeneity of cancer. One example of multiscale modeling is radiogenomics or imaging genomics, where essentially the goal is to look at medical imaging, such as MR, CT, PET images of tumors, and using what is called radiomics to analyze these images. And secondly, to link that information with genome-wide data, for example, gene expression data, copy number data, methylation data, any omics. And the goal, why are we doing this? Why are we doing multiscale modeling and radiogenomics or imaging genomics? The idea is precision medicine, is essentially to develop models specifically for a patient and enabling patient-tailored therapy. Essentially, this is a data-driven medicine where we are using molecular data, cellular data, and imaging data, and clinical data to make these models. Here are some definitions before we go forward. In the beginning of the talk, I will present some work that we did a couple of years ago, where we essentially use radiomics or a more traditional computer vision methods that essentially are defined as semi-automatic extraction of quantitative image features from medical images. These are essentially algorithms that allow us to extract hand-defined features from tumors and lesions. And these features capture edge sharpness, texture, intensity, and they also allow to describe the environment of the tumor, both globally and around the lesion. And then secondly, the more recent approaches are essentially based on deep learning, convolutional neural networks, where we learn these features directly from the data. We don't have to define them. And of course, this is more black box modeling. And so what are our goals? First, we want to study the fundamental molecular biology behind multiscale phenotypes. So we want to see, is there any cellular or tissue signature of a lesion? And these signatures essentially become additional phenotypes to link with. And we want to know what is the biology causing these phenotypes. And secondly is essentially the opposite direction, where we're using the imaging to predict molecular information. Because that essentially gives us non-invasive biomarkers for biomedical decision support, and this allows diagnosis, prognosis, and treatment response predictions. And we can do this either alone or integrated. So these are the three projects that I want to talk to you about. First, a more general overview of the radiogenomics mapping efforts that we have done in the past, and essentially the concept of the radiogenomic map. Then I will discuss unsupervised analysis of brain tumor imaging and genomic data. And finally, I will discuss some validation of these radiogenomics maps and how we use also deep learning. So first, I'm going to explain our framework that we have developed for radiogenomics maps. And is we essentially have developed these methods in non-small cell lung cancer and glioblastoma. So what is this radiogenomic map? Essentially, these are statistical associations that we're modeling between imaging and molecular data. And we essentially want to learn more about the biology of imaging phenotypes. What is the molecular biology? What are the genes and the pathways that are responsible for certain image features? And again, the idea is also to look for non-invasive biomarkers. Is there any image feature that represents an actionable drug target, for example? So there's many different ways of integrating imaging data and molecular data. We can look at univariate methods and look at correlations and assess significance. We can build supervised models of image features in terms of genes and metagenes, and genes in terms of image features. And we can also look for multivariate associations between genes and image data. First, I need to explain to you how we are essentially extracting these features from medical images. So here's an example of a lung tumor and a brain tumor. And essentially we have developed at Stanford with collaborators a platform that allows radiologists to annotate these tumors, annotate features, and also provide us with segmentations that we then can use for extracting computational features. So this is this tool, which is called ePAD, and this tool allows segmentation and annotation. You can see here some examples of some features that characterize lung tumors. And secondly, the tool also allows essentially to annotate lung cancers and to segment these lesions by determining regions of interest. Once we have these segmentations, then we essentially can define features, and we essentially have three types of features that we extract from these lesions. One class of features are texture features, such as Gevaert filter banks. We also characterize the shape of the lesion. This essentially gives us an idea of how the shape evolves around the edge. And finally, we also look at the edge sharpness, which gives us an idea of how the intensity changes from the inside of the tumor to the outside. And then we essentially are able to define lesions that have blurry edges or sharp edges. So overall, we can extract hundreds of features from these images, and from both CT images and MR images of, for example, lung and brain cancers. In the case of brain tumors, we actually have multiple regions that we segment from these tumors. We look at features of necrosis areas, enhancement areas, and edema areas. So then, in this case, we have essentially 3 times the amount of features because we can characterize all these different regions. So now, how are we developing these radiogenomics maps? We essentially look at molecular data, for example, gene expression data that gives us an idea of the activity of each gene in the human genome using RNA sequencing. And in the case of glioblastoma, we use the dataset from the Cancer Genome Atlas, where we have essentially data of 400 patients. We summarize these gene expression data into gene expression modules using bioinformatics algorithms that I won't have time to elaborate on. And then once we have these modules, we essentially can then link these modules with image feature data. So this is essentially our overall idea of radiogenomics map. So we have our images, we extract features, we have our image features matrix, then we have our gene expression data, which we then use dimensionality reduction methods to reduce the dimensionality because we don't have enough data to link single genes with image features. And then we develop statistical approaches, supervised machine learning approaches to link image features with metagenes. And then finally, what we also can do is we can link these associations and this radiogenomics map with other public data. There are many databases, for example, GEO and ArrayExpress, that provide gene expression information from other patients. And what we can do is we can actually link these metagenes across these cohorts, and then indirectly we can link essentially the association of metagenes with image features with other clinical outcomes. And this essentially allows to use the radiogenomic map to link image features of new technologies through gene expression data with, for example, treatment outcome. So this is the work that we did initially in radiogenomics maps. And this statistical framework essentially allows us to map image features to metagenes, correlate these image features with survival using public gene expression data, and also develop multivariate survival models. And we have demonstrated this work in lung cancer and glioma, and we are ongoing work also in head and neck cancer. So now, in a second project, we essentially flipped this around, and we decided to focus on the imaging data of brain tumors and using unsupervised radiomic analysis. The idea in this project is that we want to see if imaging signatures exist in gliomas or glioblastomas that can be used to inform treatment decisions. So in this project, we essentially have three steps. First, we investigated if these image features that we extract from tumors, if we can use them to identify subtypes of glioblastoma using this radiogenomics approach and using clustering algorithms. Then we will validate these subtypes. And finally, we're going to associate these subtypes with molecular pathways. And again, essentially, we are using radiogenomics here and molecular interpretation of these subtypes. So this is a visual way to represent this workflow. So first, we have our multimodal MR images. We extract image features, and we are essentially aiming to discover these subtypes. Then we validate these subtypes in a second cohort. And finally, we're going to associate these subtypes with activity of molecular pathways that then allow us to associate this with drugs. So first, we used a Stanford cohort of glioma patients to develop the subtypes. So these are patients that were diagnosed between 2001 and 2010 and got an initial de novo diagnosis of glioblastoma. And virtually all patients were scanned on the same scanner. And this allowed us to select about 360 patients. But we further selected these patients for a subset of patients which have solitary non-midline crossing lesions, which reduced the number to 221. And then we selected images with more or less the same scanner characteristics. So now we can use this quantitative image feature pipeline that I described in the first part, which is often called radiomics. And essentially, we use these two images, T1 pre and post contrast images. We had radiologists annotate regions of interest for these tumors and essentially represent these tumors using these image features. These are some examples of some tumors that have been outlined from the Stanford cohort. So we used essentially our radiomics pipeline, extracting these intensity features, shape features, and edge sharpness features to characterize these lesions. So now what we want to do is once we have discovered these subtypes, we want to validate them in a second cohort. And in this case, we can use the cohort from the Cancer Genome Atlas. So we use the same framework to extract features from the second cohort, but it's important to mention that this cohort is much more heterogeneous, so it comes from 4 centers, and there's all 3 main scan manufacturers are involved, and of course the scan parameters are also much more heterogeneous. We had initially 193 patients, but if we selected only solitary non-midline crossing lesions, we reduced the number to about 145 cases. So first, the unsupervised analysis on these cohorts. We use consensus clustering. This is an algorithm that is very well known in the bioinformatics community and has been shown to be very robust in developing subtypes. The idea behind consensus clustering is that you don't cluster the data just once, but you sample from the data, and you sample hundreds or thousands times, only a subset of the data to only discover subtypes that are robust across all these iterations. So we tried many different numbers of clusters from k equals 2 to k equals 6. And based on statistics of the data, the solution with 3 clusters was the best solution. Next, we wanted to figure out what is the meaning of these subtypes. Can we interpret these subtypes and in a way that we can actually visualize it? And we used a modified t-test, which is called significance analysis of microarrays, to verify which image features are essentially characterizing each of these 3 subtypes. And this is a visualization of the results. So here you can see in the rows, these are image features that were extracted from these patients, and the 3 clusters, and how important each feature is to determine each cluster. So cluster 1 was essentially defined by features that characterize irregular edges and irregular shapes. So these are all features that are local area integral invariants, and this is a class of features that captures lesions that have very irregular shapes, so that are non-convex. Secondly, this is the feature that characterizes cluster 2 the most, and this feature is essentially compactness, which means how spherical the tumor is. And cluster 2 captures tumors that are very spherical. So essentially more or less the opposite of cluster 1. And then finally, cluster 3 captures glioblastoma that show rim enhancing, which is captured by these edge sharpness features. And these are essentially tumors that have a very dark core, and then essentially what is presumed to be necrosis. So this essentially allows us to define these 3 subtypes. Subtype 1 with very irregular shapes and many concavities, which is what we also call the pre-multifocal subtype, because this subtype has a lot of features that we saw also in multifocal tumors. Cluster 2 is essentially the spherical cluster with very regular edges and spherical lesions. And cluster 3 is the rim enhancing subtype, which is defined by blurry edges and rim enhancing. And here you can see an example, a 3D rendering of these classes. Then what we wanted to do is we wanted to investigate if there were any clinical significant difference between these subtypes. So we looked for differences in age, sex, Karnofsky score, and we found mainly tumor volume to be significantly different between these clusters, with cluster 3 capturing more larger tumors, as you can see also here. So cluster 3 has larger tumors compared to cluster 1 and 2. We also looked for any correlation with molecular parameters, such as hypermethylation of MGMT or EGFR amplification, and also tumor location, but we couldn't find strong significant correlations. So now that we have discovered these subtypes, we wanted to validate if these subtypes also exist in a second cohort. To do this, we used again a method that is common in bioinformatics analysis, and this method is called prediction analysis of microarrays. Essentially, it allows us to define a model for each of the 3 clusters based on the Stanford data, and then use this model to assign patients in the new cohorts to each of these 3 clusters. And then secondly, we used a statistical test, which is called in-group proportion analysis test, to verify if these clusters actually exist in the new cohort. Here you can see the results of the IGP test. So essentially for each of the 3 clusters, we could show that the p-value of this test was very significant, and the in-group proportion statistic was high. And essentially we saw also that the clusters had more or less the same proportions of patients in the TCGA cohort versus the Stanford cohort. So cluster 2 is slightly larger with 40% of the cases versus the 2 other clusters. So essentially, this allows us to validate these clusters and that they also exist in this second cohort. Next, what we did is we also looked at the clinical characteristics similar to the Stanford cohort, but we could not find significant associations. One thing that we have in this new cohort is that in the TCGA cohort, all these cases are have extensive molecular data. So we also have the proposed molecular subtypes based on gene expression data. But we couldn't find strong correlation between the 3 imaging subtypes and the 4 gene expression subtypes. We also compared and annotated these tumors, or we looked for associations with other molecular subtypes, such as SIMP, which is a hypermethylation phenotype, mutation in IDH1, MGMT, hypermethylation, and EGFR amplification. But we could not find any significant association. Finally, we wanted to map these subtypes to activities of pathways. As I've already mentioned, in the TCGA cohort, we have extensive molecular multi-omics data. And that allows us essentially to look for these tumors and for these subtypes and investigate which gene expression pathways are essentially active in each of these cases. So to do this, we used an algorithm called Paradigm, and this algorithm essentially combines DNA copy number data and gene expression data to estimate the activity of known pathways from the NCI pathway interaction database. This is how this algorithm works. So essentially, we have here a toy example of a pathway, which is a very well-known apoptosis pathway between MDM2 and TP53. Essentially, what this algorithm does, it uses a Bayesian model to estimate these activities of these genes and how the pathway is active using these two data sources. And then essentially what we get as a result is a pathway activity matrix with the pathways in the rows here and the patients in the columns, where we can essentially assess the activity of each pathway, where red means, for example, that a pathway is very active, and blue means that the pathway is inactive. So now what we can do is we can, again, associate these pathways with these three subtypes and figure out which of these subtypes essentially activates which pathway. And this is what we found. So essentially we found that cluster 1 upregulates the c-KIT pathway. Cluster 2 downregulates many pathways, including these three. And cluster 3 upregulates the PGGFR pathway, VEGFR pathway, and many other pathways that are significantly upregulated. So now we can essentially add this annotation to our three subtypes. And so essentially the pre-multifocal subtype upregulates one pathway, cluster 2 is downregulating many pathways, and cluster 3 is upregulating many pathways that are targeted by drugs. So in the case of cluster 1, this suggests that this cluster could be treated with imatinib or dasatinib, which has been shown to have activity against c-KIT. So this is a known c-KIT tyrosine kinase inhibitor. And finally, also for cluster 3, several drugs could be suggested based on this. Bevacizumab, which targets the VEGF receptor, and then also a number of anti-angiogenic multi-kinase inhibitors that target VEGF and the PGFR pathways, sorafenib and sunitinib. So essentially, taking this all together, these imaging subtypes provide ways for non-invasive biomarkers to assess treatment. Finally, what we also did is we investigated the survival correlation of each of these three subtypes, and we found that cluster 1 essentially has a poor survival, which you can see here, and cluster 2 has an intermediate survival, whereas cluster 3 has the best survival. So to summarize this study, we showed using unsupervised analysis that we identified essentially three stable clusters. We developed a model then to validate these clusters in the TCGA dataset, and we investigated which pathways were activated by each of these subtypes. So now we essentially are going to investigate, can we validate these pathways and can we associate, can we prove that, for example, in cluster 3, that bevacizumab is more effective in this subgroup. So what I've just shown you is that this group 3 just activates this VEGF pathway and many other pathways. And several studies actually have tried bevacizumab as a first-line treatment in de novo GBM. But they showed that it did not improve overall survival. It did improve progression-free survival. And some reports also show that quality of life improves with this drug. So the idea is now that we want to validate the subtypes in a large cohort from a recent trial that was run by the Radiation Therapy Oncology Group, and a trial that was evaluating this bevacizumab as a target of group 3. So we want to see what is the response in group 3 versus group 1 and 2. and essentially prove this idea of non-invasive diagnostics for treatment allocation. This is the trial that was done. And essentially in this trial, 637 patients were randomized with 312 patients receiving the drug. The trial was run by this group, and we have requested the multimodal MR data, but this data is of course very heterogeneous. There's no standard annotations, and we have no manual, we have no segmentation. So we need to segment these lesions to figure out to be able to extract the image features and to be able to validate our model. Therefore, we essentially needed a better solution than manual segmentation. And there's a well-known challenge that has been organized in the past couple of years by BRATS, by the Brain Tumor Segmentation Challenge, and this is organized by Mikhail. And essentially what they have done is they have provided multimodal MR image data for over 220 samples in the most recent challenge, and they provide 4 MR modalities. All these patients have been pre-processed and standardized, and expert segmentations have been provided for based on 11 radiologists. These radiologists have essentially annotated these different subregions of these tumors, and this is a visualization of these subregions. And typically, and the idea of this challenge is that groups around the world can submit algorithms to predict segmentations using only this multimodal MR data. The way that these algorithms are evaluated is using the DICE score, which is essentially the harmonic mean of the precision and recall, and gives you an idea of how well your algorithm is actually capturing the segmentations of radiologists. So, so we essentially wanted to develop an algorithm to do this and use this algorithm on the RTOG data. Because our problem, of course, in this cohort is that this data set is not annotated. We also need to identify the correct modalities. One big issue in the community is that segmentation algorithms do not generalize well to new cohorts because of the fact that the data is very heterogeneous. And also, in many cases, not all modalities are available. So therefore, we started to develop a method for automated brain tumor segmentation. And we used essentially convolutional neural networks to solve this problem. So we developed essentially a model that uses 3D images of the brain, multimodal images, and our goal is essentially to predict these 4 classes. So we essentially have a background class, and then we have 3 foreground classes, which is essentially edema, necrosis, and enhancing tumor. We used a convolutional neural network, which is a VNet model, which is very popular at the moment for segmentation tasks, especially also in medical imaging. And one of the key features of this model is that there is a contracting path and an expanding path. So in the contracting path, using repeated convolutions, the image is condensed and then expanded again. And one of the key features is that there's also these concatenation links that essentially concatenate layers across these 2 paths. I'm not going to go over the details of the training of the algorithm, but we used the BRATS data to train this algorithm with over 285 patients, of which 210 were glioblastoma and 75 patients were low-grade gliomas. We had the axial view of the brain, and we had these 4 MR imaging modalities. And essentially the goal is to predict these 4 classes that I mentioned before. Here are the results. So we are essentially predicting 3 tumor volumes, whole tumor, tumor core, and enhancing tumor. And here you can see the DICE scores, where essentially DICE score of 100% would mean that we exactly reproduce the pooled annotations of the experts. So here you can see that for whole tumor and for all patients, we have very good performance of almost 90%. And then we also look at how the algorithm works specifically on glioblastoma versus low-grade glioma, because of the fact that many low-grade glioma do not show enhancement, we typically see a lower performance for tumor content enhancing tumor in low-grade gliomas. Next, what we wanted to investigate is which MR imaging modalities are actually essential for this tumor segmentation. So here I'm showing you the same result again as before, using all 4 imaging modalities. So next what we did is we dropped the T1 pre and the T2 flare modality, and we found that essentially the results did not change dramatically, suggesting that these 2 modalities are not important. Secondly, when we dropped the T1 post and the flare, we actually saw a dramatic decrease in performance. For example, when we dropped the T1 post, we had a very significant drop in the performance for tumor core in enhancing tumor. And when we drop the flare, we have a significant drop in the performance for the whole tumor segmentation. Secondly, we can also drop 2 imaging modalities, and essentially suggested by the previous experiment, when we drop the T1 pre and the T2, we essentially still have a very good performance compared to using all 4 modalities. Whereas, of course, when we drop the T1 post and the flare modality, we saw a very poor result for certain tumor volumes. So taken together, this essentially suggests that we only need the T1 post and the flare, and this is important, especially for retrospective cohorts, because we typically do not have all imaging modalities in these cohorts. So what we have done now is we have essentially built models and evaluated them on the same data set. And as I mentioned before, people have shown that it's very hard to generalize these models to new data. So, and this data set is essentially very idealized data because it is heavily pre-processed, skill stripped, and multimodal registration was done, and also the resolution was matched by interpolation. But what happens when we apply this model to a new data set where these steps have not been done or not in the same way? So we essentially found a second cohort, which is the Rembrandt cohort, which is actually an older cohort than the TCGA cohort. And we were able to collect 57 patients that have multimodal MR images. The volume size of these images is different. We also had the actual view of the brain, and we had the 4 imaging modalities. And then we had 3 radiologists essentially develop a consensus annotation of these tumors to essentially collect ground truth. So these are the 2 issues that we had to solve. First, the image sizes are not the same between these 2 cohorts. And essentially, we evaluated several ways of interpolation, but essentially the simplest interpolation, linear interpolation, gave the best results. And secondly, also the image intensities and the intensity distributions were very different between the 2 cohorts, between BRATS and between Rembrandt. And our solution is essentially fine-tuning, whereby we use a subset of the new cohort to keep training the model. And I will explain that in the next few slides. So these are the 4 experiments that we did to essentially show or test if transfer learning is working. First, we have, as a baseline, we just have a random initialization of the weights of the convolutional neural network. We essentially did no training just to figure out the baseline. A second experiment is doing pre-training, where we essentially develop a model on the BRATS dataset and then immediately apply it on the Rembrandt dataset. Then the third experiment, which is this retrain experiment, then we essentially have no pre-training, and we train the model on the Rembrandt dataset and also test it on that dataset. And the final experiment is essentially the transfer learning experiment, where we train the model first on the BRATS dataset and then uses 5% of the new dataset, of the Rembrandt dataset, to fine-tune the model. And here are the results. So as we expected, when we use a random initialization of the weights, of course, we have a very poor performance of predicting these segmentations. When we apply the model directly from the BRATS to the Rembrandt dataset, we actually have already quite a good performance. So we have, for example, for the whole tumor, we have 83%, we have a dice score of 83%. This improves when we do retraining slightly. So when we train on the same cohort and test on the cohort, we essentially get this performance. But the best performance was essentially observed when we did transfer learning and used the model from the BRATS cohort and then tested it on the Rembrandt cohort. So to summarize, so this segmentation project essentially taught us that we only need 2 modalities, and there's only a minor drop in the performance when we only have T1 post and flare images. And this is important, again, because in many retrospective cohorts, we don't have all these modalities available. And secondly, we suggest that fine-tuning is a good way to do transfer learning, where we only use a small set of data from a new cohort to get the best performance. So our ongoing work now is to use this automated segmentation algorithm together with the pre-processing pipeline on this RTOG data set, and then eventually test our hypothesis that the survival in subtype 3 is better than in subtype 1 and 2. So to summarize our work, we have developed a radiogenomics a mapping framework, a statistical framework to associate molecular data with MR imaging. And we've tested this in brain tumors and lung cancers. And essentially, we are focusing on using this framework to study clinical outcomes. Our most promising application is really non-invasive precision medicine. And finally, we want to use MR imaging as a biomarker for treatment decisions and treatment response. These are the people I have to acknowledge for this work, and of course our funding. I thank you for your attention. I'm happy to answer any questions.
The question actually, it was pretty impressive, experience that you provided us. I think overall you're at Stanford, well-curated data is essential for good quality output. So how do you deal with heterogeneous data? I mean, it's a difficult question, especially when you deal with large retrospective data sets. How do you select the data? What's your criterion to say, this one is out, this one is in, and if it's in, what are the criteria for that? So that really is a major issue, I think, with radiogenomics and radiomics data.
So in the GBM cohort, in the first part of the, or essentially our GBM cohort that we have collected from Stanford, that's exactly what we're doing, right? So we're trying to figure out what is the set of patients that is the most homogeneous as possible, scanned more or less with same scanner characteristics such as slice thickness, pixel resolution, scanner model, and things like that, and start from there. And of course, when you limit yourself to a single institution, then more or less you have relatively homogeneous data. It's only when you essentially collect data across different institutions that, of course, we have more issues and more heterogeneity. So, and that's something that we observed in the first story, that when we flipped the two cohorts, so when we did the unsupervised analysis first on the TCGA data and then on the Stanford data, it did not work. Because then we are learning from heterogeneous data and testing it on homogeneous data. So the heterogeneity was too large to find any reproducible models. So the lesson from that is really that you need homogeneous data to start with, for sure.
And the other quick question also, obviously providing association with gene expression profiles is an interesting one, although it's almost comparing apples and oranges because the different processes we're looking at phenotype with imaging, we're looking at something at genetic level, but, and you're looking at the end, the whole goal of this, of course, it's probably important. What's important is really to predict outcomes, right? And then, so the question is, can we just bypass genomics and just go and try to just work more, focus our effort on predicting outcomes with radiomics instead of having that additional step of radiogenomics?
So there's two aspects here. First of all, definitely we can go directly to outcome, and many people have done that and have shown that imaging can be used to predict outcome. And we have ongoing work using CT images of lung cancer patients, for example, that shows how we can predict outcome directly from the images. But the molecular data, linking the imaging data with the molecular data has other applications. For example, looking how much of the biology can we predict from the image. And we have shown that definitely portions of the biology you can predict. And other groups have shown this too. For example, in lung cancer, many reports have shown that you can predict EGFR mutations from the image. And this has important implications for therapy monitoring, for example, because then you can use imaging to follow maybe resistance of anti-EGFR drugs, for example. So I think both applications are things that we focus on, because from the machine learning side, we don't, our models are essentially the same, whatever we want to predict. So we are interested in both of these applications.
I have a question. how do your image segmentation affect the future extraction? For example, if the edge feature, if you segment maybe change a little bit, maybe the edge feature maybe change or not.
Yes, so we have studied the robustness of features in lung cancer and liver tumors. And essentially, what we do is when we have radiologists annotate tumors, we look at variability across radiologists, and we eliminate features that are too variable across segmentations. But going forward, we assume that we will use more deep learning algorithms to automatically segment lesions. And with these algorithms, although maybe it's hard to tell if the segmentation is the truth, but it will not vary, right? So the model will always make the same decision in the same, given the same data. Essentially, we'll combine these segmentations together with the original images then to predict any outcome of interest, for example, survival or molecular data.
Thank you.
So thank you so much, and very nice work. So, I believe that I missed the last point. So now, were you able to replicate the subtypes on the trial data? What is the status there?
So we are still processing this RTOG data. So it took us about 2 years to get the data first after requesting it. We actually still haven't received the actual outcome data of these patients, so we don't know which patients were treated. We are now doing the segmentation work on this cohort.
Mm-hmm.
But we will definitely be able to assign patients to groups. There's no difficulty there. The question is, of course, do we see a survival difference?
Right.
That's our main.
Right. So concerning the segmentation, would it make sense to just use some semi-automatic tools? I know
Sure.
that I'm going exactly in the opposite direction of this meeting, but just for the practicality to focus on the subtypes, because I believe that's a very, very important topic, and ultimately the number of data sets is not that large in this case. The data is very precious.
Mm-hmm. So there's a couple of things. So first of all, the reason why we developed an automated segmentation algorithm is actually because it's very cumbersome to have radiologists annotate these lesions. So actually, during the process of requesting the RTOG data, we were able to develop this algorithm, which has comparable performance to what we have seen in the literature. It's essentially just to save time. Because it's much faster, we can process more images in a faster amount of time. And I forgot the second part of your question.
So that was the answer. And one more. But to me, the resulting errors were still quite, I would not say substantial, but perhaps prone to impact the results, right? The classification results and so on, no?
That's definitely true, but keep in mind that segmentation is not our final goal, right? So our final goal is to predict treatment or to predict survival. Right. So maybe it's not needed to have the true segmentation. And does the true segmentation exist, right? So the only thing we're doing is we're comparing with the radiologist, but maybe, yeah, where is the actual border of the tumor? So the question is how accurate do you need to be able to use it for precision medicine?
Thank you so much.
We have time for one more quick question.
I'll try to be brief. I work on preclinical imaging, and we'll need the same kind of tools to analyze the effect of tumor treatment. So there are two things that you mentioned that caught my attention. The first one is you are using Avastin, so an anti-angiogenic, and then you are using post-contrast, and you're suggesting that post-contrast is one of the images that will be useful for segmentation. So one freebie is dynamic contrast enhanced MRI. Fortunately, it's at a low resolution. If you were to add it, do you think you'll be able to add more strength, and how do you address the difference in resolution?
So definitely we want to add more functional MR modalities. The only reason why we haven't done it is because in these cohorts that we have, then the number of patients drops. And that's the only reason why we haven't done it. But we are prospectively collecting more patients with diffusion imaging, perfusion imaging, and so on, depending on what modality we're looking at.
Perfusion is 2 minutes or 5 minutes. And you have to wait anyway if you inject contrast agent, right?
But so
So
we are limited by going to retrospective cohorts, because for these type of models we need hundreds of patients,
get
right?
that.
So if we prospectively start collecting, it will take us a few years before we have a decent cohort. But definitely we expect using multimodal image, and we have done this, for example, in rectal cancer, where we have diffusion imaging together with anatomical imaging, and we see that diffusion is a very important modality to predict treatment response.
So how do you deal with the anisotropy? Because with dynamic, you're gonna be voxel looks like a
we
fry,
don't
right?
we don't necessarily need to link the modalities together, right? So we suppose we do a radiomics approach, like just a feature extraction approach. Then we just extract features from each modality independently, and we use them in our supervised models.
Thank
right.
you.
Thank you very much for that outstanding talk and for the interesting discussion. I'd like to present you with this token of our appreciation.
Thank you.
Thank you.